Ethan Zappich, growing and breeding mushrooms at home.
All right, hello everyone. Thank you so much for having me out at Nerd Nite today. I'm stoked to deliver my presentation for you about growing and breeding mushrooms at home. Sorry if I sound a little nervous, I'm still shaking off the last presentation. I've never actually had every single slide be full body chills. Like we started with a horror presentation, I did not expect that. The fish tongue isopod one really got me, anyways. I'm gonna stop talking about that. We also have some helpful tips to avoid a zombie apocalypse. We'll need some of those. Like was mentioned, my name is Ethan Zappich. I'm a director for the Alberta Mycological Society. I'm also a complete mushroom nerd. I promise I qualified to be here. That can be further evidenced by the fact that I have a mushroom and nature-related YouTube channel. I've been learning about mushrooms and related content and growing them for about 5 years now. So have amassed a little bit of knowledge. I don't feel like I'm much better at it than when I started, honestly. But why mushrooms? Why do I even like mushrooms to begin with? That's a good question that I'm glad I asked. It's because mushrooms, like any nerd would probably say about their respective niche, mushrooms, I like them because they're so freaking cool. Edible mushrooms, you can eat so many. Poisonous mushrooms, some will just kill you, right out. Other mushrooms that you can take and have a one-on-one visit with God. You can use mushrooms to make dyes. You can use them to make clothing. You can use them to grow building materials. The list goes on. Some are pink. Some are, oh no. I've done something wrong. I will hit the button. All right. Some are pink. Some are blue. Some are green. And we have to give credit to that one because it's not easy being green. I love mushrooms because they are so diverse. They do so many different things, and they were a great excuse for me to better connect with nature. Because every single time I went out, I had something new and exciting to look for. And I naturally, as I started foraging, began getting very curious about what are all these different mushrooms. And as I got better at identifying those different mushrooms, I got a lot better as well at identifying the edible mushrooms. So everything you see on screen right now is an edible mushroom, pretty common ones that you can find locally. We have the crown-tipped coral up here. We have some puffballs down here, mica caps, scaly hedgehogs, and we have coral tooth, which if you know of any of the more popular mushrooms like lion's mane, this is kind of Alberta's lion's mane relative, my favorite edible mushroom. And of course all these mushrooms are really cool, but the big problem I kept running into when I would find one of them was I could never find enough. They're not like plants. You can't come back year after year and have a big harvest. So I started looking into growing mushrooms. Before I dive into the nitty-gritty, I'm going to give you a brief overview of what the whole process of growing mushrooms is, kind of in a nutshell. So the whole process is gradually giving your fungal organism bigger and better food sources until it has enough nutrients and need to fruit mushrooms. Because mushrooms are just the fruit of the mycelium that we're actually working with, the organism, like an apple would be to a tree. It's just the way that they spread their spores and continue the life cycle. So the whole process starts on agar, which is like a gelatin kind of compound. It's derived from seaweed. And we start the mycelium on there, and it grows on a nice, easy kind of two-dimensional plane. So once you get a beautiful looking rhizomorphic mycelial culture, they don't all look like that. That's an especially pretty one. And just to give you a little more info, agar is really good for identifying contaminants as well. So any bacteria, things that shouldn't be on there will stand out really well, and you can make sure you have a clean culture before moving on to the next step, putting it into some grain, something that has a lot higher nutrient density. So in this jar here, I typically grow on popcorn kernels. So we have some sterilized popcorn, some vermiculite, some corn syrup. This is known widely in the home mycology space as drippy corn. So that's what I grow on. And once it grows out on there, we can put it into our substrate. And the substrate, does anyone know the grow your own mushroom kits that you see around where you just cut them open and they grow on your counter? That's all that this is. So it's the grain spawn, once you put it into your substrate and it grows out and colonizes, that's what that is. So the block here that you get gives you a ton of different options depending on the species you have. You could use it to inoculate logs outdoors. You could plant it in your garden and fruit mushrooms there. What I usually do is I'll put it into my grow tent, which is a humidity, temperature, and air-controlled space, a very clean space, and I will fruit it. So here is a local wild oyster mushroom that I managed to clone from the woods outside my house and start growing there. This is about a 30-hour time lapse. They grow incredibly quickly, and once I pick off those mushrooms, they'll just grow right back. So I'll get 5 or even 6 flushes out of each block. It's a really good payoff. We were talking about insects as a food of the future. I really think mushrooms are going to be a big one too. So when I say I cloned the mushroom from the woods outside, what does that mean? It means I literally cloned it. So what you can do is you can take a sterile piece of any mushroom and you can put it on an agar plate, and if the agar solution is correct, but there are some good general ones, the mushroom will grow out, the mycelium will grow out, and it'll be a direct genetic clone of the parent mushroom. Pretty cool. The other way that we can collect mushroom genetics is from spores. And so you can take a spore print of a mushroom by putting its cap on a piece of paper or tin foil, and it'll release its spores, and you can use those to germinate mycelium as well on an agar plate. This will have much greater genetic diversity, and it won't be a direct clone, like the offspring of humans or dogs, or insert any living organism here. What is the catch? Again, I'm glad I asked. Sterility. It's all got to be done under extremely sterile conditions. So to give you that same example, in cloning a mushroom, the agar plate has to be 100% sterile, the piece of the mushroom you take has to be 100% sterile, and the scalpel blade has to be 100% sterile, as well as the workspace that you do it in. Got to be very, very clean. And the reason for this is we're providing the perfect food source for our mushroom culture to thrive, but we're also providing the perfect food source for any other contaminants to thrive. So we got to be really careful. As I started getting further down this rabbit hole and wanting to grow more and different mushrooms, I found out that I need a lot of gear. I don't live in an industrial lab. I live in my home, and this is home mycology. So I had to build out a mushroom lab kind of in my house. This is my setup. If anybody wants some negotiation lessons, let me know, because I don't entirely know how I talked my partner into this one, but I must be decent at it. To give you a quick rundown, we have my grow tent cut off on the side there. I've got a fan attached, a humidifier, a smart controller to automate that temperature and humidity. I have a fridge to store my mushroom cultures. I have a dehydrator, a pressure cooker, a single burner so I can run that in the lab. I have a laminar flow hood, a microscope. None of this happened overnight, but I just kept kind of tripping further down the rabbit hole and ended up getting a lot of different stuff. But the backbone of all this work is the pressure cooker and the laminar flow hood. So the pressure cooker lets you sterilize all these different media, the media that you're using. I can get up to 15 psi, which is just over 120 degrees Celsius, and at a certain amount of time in a steam sterilization cycle, you can sterilize the different mediums you're using to grow out the mycelium. The laminar flow hood is the only place that I will then open up those mediums that I've sterilized. So in the pressure cooker, I can put some different, I usually use mason jars as the containers because that glass holds up really well in the pressure cooker at high temperatures. I'll poke some air exchange holes and put a filter patch on them so that when I take it out, everything inside is sterile except no external contaminants can get in through the filter patch. I will open it up in a laminar flow hood, and what this is basically a simple device that just has a fan in it and a HEPA filter on the front. HEPA filter is a filter that filters at 99.97% efficiency of contaminants. So it gives me a really good chance that everything I'm working with in that space is going to be clean, and I'm not going to get kind of random spores floating in there. So here is just me doing some work in front of it, sped up, of course. I don't work quite that quick. And yeah, I'll just chop this up, put it in the grain jar, and then I'll shake that around, and the mycelium will have a really good starting point. Okay. You must be thinking, wow, that is expensive to get started in growing mushrooms. But it actually doesn't have to be. Fortunately, with the advent of the internet and a very innovative home mycology crowd, people have found ways to do this a lot cheaper. And this is kind of where I started. You can use an Instapot as your pressure cooker. It gets up to 12 psi. So even though it doesn't do a proper steam sterilization, because you need it to get to 15 psi, people have really good results just raising their sterilization times. For example, if I was doing agar plates at 15 psi, I'd do them for about 30 minutes. I could do these at 45 minutes. The other thing, you can replace that laminar flow hood with a plastic box with some holes cut in it that you wipe down on the inside with alcohol. This is called a still air box, and it can get you pretty far as well. So there are much cheaper ways to go about this. And depending on the species that you're growing, if you have a really aggressive species, you actually don't need to sterilize everything at all. Oyster mushrooms are a fantastic example. Here I am taking a spore print of some oyster mushrooms growing out of a bucket, and I did not sterilize this whole bucket in my pressure cooker. I just filled it with hot water on some pasteurized wood chips that you would get for a barbecue and put the oyster grain spawn in there. Oysters are so aggressive and so contamination resistant that they'll just grow out. So I grew this and fruited this in the garden this year, and it would fruit every couple weeks, anytime it rained, and I would go pick off the mushrooms, which was pretty good. So this technique in the mycology community is called bucket tech. In the mycology community, tech just refers to technique. If you develop a new way of doing a certain process, then you have made a new tech and you get to name it. Very innovative name on this one. So just as an example of how aggressive oysters really are, here is an oyster mushroom fruiting out of my agar plate that I had left just a little bit too long. I thought that was pretty cool, and they are seriously so aggressive. I left, like I said, this one a bit too long. It's something new I'm trying out. It's called neglect tech. And that leads us on to the next point. Mushrooms don't care about your feelings. And this one's really important. I'd really appreciate if everyone could repeat it with me on 3. 1, 2, 3. Mushrooms don't care about your feelings. No, they seriously don't. These aren't plants, like I mentioned before. They're not going to be consistent for you. They're not going to do what you want when you want them to. If I could compare it to anything, I heard someone else say that mushrooms are more like cats and plants are more like dogs. It's not that you can't kind of corral them, but they're going to do their own thing at the end of the day. Oysters are so aggressive that they'll even grow on toilet paper or anywhere else. And it's really easy to think when you start growing mushrooms that you're the best grower in the world when you start on these really easy species like lion's mane and oysters. They'll grow anywhere. But as you start exploring and expanding with different species, you might start running into some problems because mushrooms don't care about your feelings. So here is a video that I've been working on for over a year now, or almost a year, getting there, because I tried to grow a mystery mushroom. I had this set of genetics that a friend gave me, and they had not included a label on it. And instead of contacting them, I thought, oh, wouldn't this be fun if I just tried to figure it out and grow this mystery mushroom? I'll try a bunch of different substrates. And that's what I went for. But about a month into the process, I came back to find my substrate bag completely filled with liquid, and I didn't entirely know what to do.
My actual best solution is going to be to poke the bag, drain the liquid, and then tape it up. But I hate that idea because it opens it up to more contamination. I don't know, it's not ideal.
And you know what? The idea wasn't so bad, but the execution was just horrendous.
Come on, drip!
Welcome to the next hour of my life, where I fight tooth and nail with a plastic bag full of mushroom urine.
Look at this space potato. I don't know what to do.
This was not my favorite Tuesday evening, and somewhat alarmingly, also not my weirdest Tuesday evening.
That sucked. I lost the holes where I poked.
So, so for the next 6 months, I continued trying to grow that mushroom with absolutely no luck. I forgot about the project. I said, this isn't going to happen. I scrapped the whole idea. I left my last culture jar of grain spawn in the fridge, forgot about it. I came back 6 months later. This is just about a month ago. And it's fruiting mushrooms in the fridge. I had tried everything before this, And this is another great example where less is more. Mushrooms will do what they want. Neglect tech. You should really try it out. So there's a lot to cover on this topic. We're just going to jump right over. Now we're going to talk about breeding mushrooms. So yeah, like I said, don't have much time. Just a high-level overview. When a daddy mushroom and a mommy mushroom love each other very much. Oh no, sorry, that's not it. Just like breeding dogs, just like breeding plants, you can breed mushrooms. You can have two parent mushrooms and isolate for certain genetic characteristics that each one of them have and kind of smush them together. Something really interesting for me is that all domestic dogs come from the gray wolf, Canis lupus, even Chihuahuas. That is an uncomfortable thought. Gives me kind of the same feeling as the bug presentation a little bit. And the same thing with mushrooms. If given enough time, you can get some really interesting expressions in those mushrooms, even within the same species. So just to give you a very brief overview of the process, what you're trying to do is you're trying to find two monokaryon cultures, which just means having one nucleus, so they have not mated yet. And you are trying to put those two from two different parent mushrooms on an agar plate and have them grow together and see if they're compatible and they want to just breed into each other and clamp together. Now, if they are compatible, they'll smush in the middle and you will have a new strain of that species. If they're not compatible, they will grow right up to each other and make a physical wall in the middle and start growing the other way. Pretty interesting to see. You can tell, again, if a culture is monokaryotic if it has these little clamp connections on the mycelium strands here. That would mean it's dikaryotic because it has already mated at each of these points. It has fused together. Okay, so now that you know how to breed mushrooms, of course it gets a little bit more intense than that. You have to dilute the spores down and try to isolate those monokaryon cultures, which can be a difficult process, very time-consuming. But it can be done, and it's something that everyone here can easily wrap their head around, I think, if given the opportunity. Some cautionary words. We have already heard about cordyceps this evening. Very interesting mushroom. The Last of Us has opened our eyes to that scary thought that, oh, there are spores everywhere, and some of them might control my brain someday. Well, this is going to be especially, oh yeah, sorry. This is something that I neglect-teched a while ago. It was one of those situations where I just kept saying, oh, I'll deal with that later, I'll deal with that later. I didn't deal with it later, and I grew something really gnarly in there. No idea what it is. And the number one thing you shouldn't do if you grow something really gnarly in your lab is open it up and release it into the world and say, here, go run. I didn't do that, fortunately. What you can do is you can put that through the pressure cooker again, and you can kill everything, and then you could send it into the world. That's probably the best thing that you should do if you find yourself in this situation. Here's something scary that's happening. Ooh. All over eastern North America right now, golden oyster mushrooms have gotten out. They're a very invasive species because home growers have just been composting them and releasing them outside, which we're told is a very good thing to do for the most part. But no, not with a tropical species that is not native to the area. So this is maybe a forager's dream to find this many tasty oyster mushrooms, but it can be very ecologically harmful to outcompete all the native fungi that are trying to decompose the forest floor. So yeah, if you are thinking about starting growing mushrooms, the easiest way is to go with something like a grow kit, but maybe consider being careful if you are growing a species that's not native to here, or you can also grow a native species like the oyster mushroom I showed you throughout this presentation. This is the aspen oyster. It's one of my favorite mushrooms, and there are lots of good local growers who can get you a mushroom block of this one as well. I hope you all enjoyed the presentation. If you would like to, you can subscribe to my YouTube channel. I do lots of experiments and all sorts of fun things like that. Thank you.
Okay, I had two quick questions. First of all, is you were talking about getting mycelium from the outside to start your own colony. but you were emphasizing how everything needs to be sterile. How do you deal with that outside?
Yeah, that is a great question. So what you want to do is you want to take that mushroom, that dirty, filthy mushroom that you find outside, into your very clean lab that you have prepared a space for, and you want to, in front of something like a laminar flow hood or in a still air box, rip open the mushroom to expose sterile tissue inside. And that's what you want to take from. Because if you were to cut into the mushroom from the outside, of course, your scalpel blade is pushing contaminants in there. But yeah, if you work quickly and you work carefully, you can rip open the mushroom, expose some flesh, take that sterile piece and put it onto your agar plate. Yeah.
Quick second question. Have you tried farming truffles, and would that make you a lot of money?
Haha, a good question. Well, I'll never expose my secrets in this public setting. No, I have not tried farming truffles. Some people are doing it successfully, but it's still not the really expensive species, and that's why they're still so expensive. I saw a presentation when I was at a mushroom convention in North Carolina that the Alberta Mycological Society sent me to in 2023. There was someone doing a presentation on truffles where at pecan farms, both retired and current, they're having a lot of truffles pop up because they associate with those trees as a major host. So if you know of any nearby, maybe go raid them with a shovel.
Okay, I have two questions. You said not all mushrooms are edible, but technically they are.
You raise a fantastic point. Let me clarify that. All mushrooms are edible at least once.
Okay, and now this is a meta question. Why, among all nerdery, are mycologists much more in favor of puns?
Also a good question.
Don't even.
I can't take the easy one, and now I'm blanking on other puns to respond with. Because we're fun guys. They're nice.
What was that one?
Your morels are low.
Oh, nice.
Because mycology is better than your ecology.
All right.
Ron was up next.
Okay, and I have a quick question too. What are you 3D printing?
Oh, I just got the 3D printer. I was explaining at the table, but I'm doing everything that I can not to 3D print toys and things I don't need because there are lots of those out there. I'm using it for different things in my mushroom lab. So right now I'm designing a kind of magnet embedded surface where I can put all my tools and it'll stick to my stainless steel table. And yeah, other things like that. Like,
Very nice.
yeah.
I saw a YouTube video about using, I think, a certain type of mycelium and growing it inside a 3D printed mold so that you can basically create objects out of mushrooms.
Yeah, that is a really cool thing. Great point. You can do that as well. And that's how people are growing, for example, mycelium lampshades and things like that. You can just print a frame and have the mushrooms kind of grow in there, and then you can dry the mycelium, and you'll have a really solid material that is still permeable and it's fire retardant. There's all sorts of benefits.
It's a biodegradable material of the future.
I've got minimal biological training, but are fungi, I've been told they're neither plant nor animal. What exactly are they in the kingdom?
Great question, Rhonda. I also don't have a biological background. I came from business and marketing and just got really into this stuff, so anybody can do it, I guess. Fungi are their own kingdom. They are one of 5 kingdoms that we have. I won't name them all because I forget one. I know I forget one. But yeah, they are an entirely different kingdom. We have flora, fauna, funga. There's something else entirely.
Prokaryotes and archaea.
Thank you.
We're at Nerd Nite.
Mentioned a little bit about feeding mushrooms. Is there any other benefits to feeding them than their protein? Can you share your favorite?
Yes, absolutely. I'm sure that everyone here has heard something about the different medicinal benefits of mushrooms. Some mushrooms have very unique, novel compounds that interact with our bodies in unique and novel ways. And Yeah, so there are some very exciting medicinal mushrooms. Even the regular edible ones, though, have so much overlap with the well-touted medicinal mushrooms. They're a great source of beta-glucans that you don't get elsewhere. They have a bunch of vitamins that you don't get elsewhere just from eating vegetables and meats. I would recommend that everybody add them to your diet in some capacity. They actually give you a lot of really good stuff. And then there are some mushrooms that go above and beyond, like coral tooth, that white spiny one that I showed you, has something that can help you regenerate nerve cells in your body. And that is an area of ongoing research. That's why lion's mane is so popular, but it has the same one.
Are these beneficial nutrients things that you can only get from a fresh mushroom, or like does a can of sliced mushrooms still have all the good stuff?
Yeah, well, the white button mushrooms will be better than no mushrooms, but there are so many cool edible mushrooms that just aren't popular, but they're getting easier to get, more accessible, that is. To answer your question, though, it's not just the fresh mushrooms. There are all sorts of ways to treat the mushrooms that can be beneficial as well. For example, I saw that exposing shiitake mushrooms to UV light will enhance their vitamin D content. Very strange, even like dried shiitake mushrooms. Mushrooms that are dried, a lot of them can be rehydrated really well, and they're almost the same consistency, sometimes better flavor once they've been dried. Yeah.
When you buy mushrooms from the grocery store, can you take a spore print? Like, can you actually propagate them?
You absolutely can.
Right.
Yeah, any of the mushrooms. Exactly. And now that we're seeing grocery stores sell some of the other gourmet mushrooms like oysters and like lion's mane in some cases, you really can take those and clone them, and you have access to a really good commercial strain that someone has taken a long time breeding and perfecting.
We talked a little bit about cordyceps in both of the later presentations there. Would I have to talk to Elon about some bugs that grow cordyceps, or is there another way to do that?
You would have to talk to Elon. I don't know a ton about the different bugs. I do know a really interesting thing, which is that it's not the same species of cordyceps that is attacking all these bugs. There's a different species that attacks all the different bugs within the genus. So they've each adapted to target that specific species of bug. Maybe a little bit more terrifying when you think of it attacking us in some capacity, that it will find a way somehow, somewhere.
Well, like, don't psychedelics modify our behavior?
It's true.
Yes.
Do they modify our behavior in a way that results in us wanting to grow mushrooms?
Yes.
Got him.
Julian, didn't we have a presentation about a year ago about somebody talking about
Medicinal use of psychedelics? Or-
yeah, they were doing a psychology presentation about how psilocybin, I think it was,
Psilocybin?
Yeah.
psilocybin changes neural pathways in the brain.
But that was a therapist that had, what's Elon Musk's drug of choice?
Ketamine.
Yeah, his major therapy was ketamine.
Yeah, okay.
Yes, there's tons of psilocybin research for therapy and tons of different stuff.
Yeah, there's lots of really exciting ongoing research.
And lots of anecdotal evidence.
Yes, lots, probably far more anecdotal evidence.
Enough anecdotal evidence becomes evidence, doesn't it?
There's a there's stoned ape theory, which is that the oldest ancestors of humanity, they first developed consciousness, and their brains started expanding at that critical point in human evolution because they found psilocybin mushrooms in the desert and started consuming them. Yeah.
Interesting. All right, I think that was I don't see any other hands. So take those hands and put them together. Thank you very much.
Thank you.